So DNA is deoxyribonucleic acid. It is a type of nucleic acid. There's also ribonucleic acid, which is RNA, but we're looking at DNA here. So deoxyribonucleic acid, the deoxy part comes from the sugar that is made up of. Deoxyribose sugar is the sugar that makes up DNA. Ribose sugar is the sugar that makes up RNA, ribonucleic acid. So this nucleic acid is DNA. DNA forms two strands. It uses two strands and it forms this double helix type where you get this corkscrew type formation. It's a very stable structure, and that's good because this is our instruction manual, right? This is our recipes. We want it to have good instructions for us. So a very stable structure is good. It is made up of nucleotides. So that's my building blocks for DNA is a nucleotide. And a nucleotide, so if you think of building something, anabolism, so I've got Legos, I'm going to put these together into this large structure of DNA. A nucleotide, one Lego, one building block is going to be made up of that deoxyribose sugar, and that's represented here by the large sugar. It's going to be represented, it's also going to contain a phosphate group that's in that small ball. And you'll notice that on the outside of this ladder, of this outside, kind of looks like a ladder, outside of this double helix, the sugar and the phosphate groups alternate. And that makes for this stable structure here. Now, extending from that sugar is a nitrogenous base. When we talked about nitrogen being a growth limiting reagent, it is made up of nitrogen. It's referred to as a nitrogenous base. And there happens to be 4 different ones that occur in nucleic acid. That is that guanine, cytosine, adenine, and thymine, GCAT. And those are represented by the different colors there. So I get 4 different types of nitrogenous bases. Sugar and phosphate's all the same. Deoxyribose sugar, phosphate, they're all identical from that. All right, so it's made up of these nucleotides put together, and you can see that the nitrogenous bases are arranged in pairs. So one nitrogenous base extends in pairs with another, and then it has that sugar phosphate backbone on the other side. This is where you get 2 strands, and then they come together and pair, meet in the middle, to create this very stable double helix. All right, what we have found out is that guanine always pairs with cytosine. So if that's represented by green and orange, guanine and cytosine always pair together. And adenine and thymine always pair together. And we'll see that in a future slide about why. Adenine and thymine always pair together. So that's something we need to know about how the structure of DNA is, that you always get these pairings. How do you remember that? I've heard students tell me cars in the garage, apples on the trees, whatever it helps you to remember that G's and C's always pair and A's and T's always pair. All right, we're going to come back to this anti-parallel strands. We know it's made up of 2 strands, but we're going to come back to this anti-parallel, but in the future. But the first thing I want you to do, the next thing I want you to do is to actually work through this problem. Now I get a lot of students that are like, okay, yeah, GC, AT's got it. But memorization's not going to cut it completely. You're going to have to be able to apply this information. So you need to practice these types of problems. So I'm going to give you a chance to work through this. The chromosome of a Mycobacterium tuberculosis. That's a familiar one. That's the one, that disease that causes tuberculosis, the pathogen, the bacteria that causes tuberculosis. We know that its chromosome, its haploid circular chromosome is 4,411,529 base pairs long. We also know a scientist isolates the nucleotides and finds out that there are 893,963 molecules of guanine. Your job is to figure out how many molecules of cytosine, adenine, and thymine are in that structure. So that's what you're going to do, and I'm going to give you a second to do that. This is something we would do in class. This is something we would work together. So call your lab group, study together. Okay, so what did you find out? And there's a couple of different ways you can approach this problem, but you needed some information that you memorized, and then I'm asking you to apply it on these questions. So you were given 4,411,529 base pairs. You needed to know what a base pair was. So we said that G and C is a base pair. Guanine and cytosine pair up. So if I were to ask you how many pairs of socks you have, you may say you have one pair of socks. How many individual socks do you have? See, those are two different questions. I have one pair of socks, but each one has two socks. So if you ask me how many socks I have two. If you ask me how many pairs of socks I have one. So we need to understand what we're talking about here. We have 4 million blah, blah, blah base pairs. Then it tells you how many bases you have. Guanine is a base, is a nucleotide. So you have 2 million blah, blah, blah of guanine. Okay, so remember guanine is a sock, and these are pairs of socks. So first thing you need to do to figure out this problem, and most of you probably figured it out this way, is that if you know what guanine is, and you know what guanine pairs with, if I have 2 million and however many molecules of guanine, then if those pair together, I have to have the same number of cytosine. Does that make sense? Guanine and cytosine have a pair. I know I have this many guanines, so that means I have this many cytosines for that, okay? So that's the first thing that's the easiest to figure out. So guanine and cytosine. So if you know the number of guanines, then you know how many cytosines you have. All right, so guanine and cytosine are pairs. Guanine is individual socks. Cytosine is another sock. Together, they make up base pairs. So to find out what's left over, what's available to be adenine and thymine, we're going to subtract this 2,893,000 number of pairs. I've got that many pairs of guanine and cytosine. I'm going to subtract that from the number of pairs I have here. And that one, I forget the number, so I have to go back to the PowerPoint. That one, when I do the math, I'm going to have 1,517,000 and 566 molecules of pairs of A's and T's. All right, so again, if I'm talking about pairs, this number plus this number is going to add up to a base pair. So that's going to be the adenine. I'm going to get the same number for the thymine, and that's going to give me what the problem asked for. And remember, the problem asked for how many cytosines, adenines, and thymines you have. So this is going to give me my answer. Now, another way I see students do it, and it just depends on how your mind works, doesn't matter. Some students will take this base pair. I have one pair of socks, and they're going to do the math to get this into individual socks. And so they're going to get 8,800,000 and whatever. They're going to do the math to get the number of individual socks, individual of nucleotides, and then all of these numbers should add up to that. You can check the math on that. So again, it just depends on how your brain works. But knowing though G's and C's pairs, again, you've got to do more than the memorization. You have to then take it and apply it. So make sure you practice that. Now, there are students, I know you're out there, it's okay, that have no idea what I just did. Go back through the video, try it again. But we work this out in class, which means work with your study groups. Figure out somebody who does know how to do it. Ask them. Work with them on Zoom, FaceTime, Go Meet, whatever you want to do, whatever you're safe to do, and figure these out. Do not let this go by and hope it doesn't come back up again, because things like this do. You need to understand it. So if this went by too fast for you, try it again on the video and then reach out to your groups. So you need to understand how to apply these concepts. So don't just blow by and say, oh yeah, G's, C's, A's, and T's, got it. And make sure that you can apply that. All right, I'm going to end this video here so that we can pick up with the next one. Good job, guys.